लगे।
It is rightly said, every revolution seems impossible at the beginning, and after it happens, it was inevitable. A very pleasant morning to everyone present over here. Our today's esteemed guest speaker is one of such well-known Indian scientists who has found solutions to many unsolved impossible aspects of the bioscience. He is a motivation to millions of people around the world. With this note, I, Dr. Dani Upadhyay, Assistant Professor, Microbiology Department, feeling honored to welcome our today's eminent speaker, renowned Indian scientist, molecular biologist, distinguished Professor Dr. Subhadip Chatterjee, sir, on behalf of entire Paro University. Sir, we are truly honored to have you with us today. I would also like to welcome Professor Dr. Girish Babu K, sir, Head of the Department of Life Science Department, Paro University Institute of Applied Science. I would also like to welcome all the faculty members and dear students to this distinctive live webinar on very interesting topic, understanding the social language of bacteria, speak or not to speak. I believe that experiencing this live webinar will enlighten your knowledge about bacteria with experimental life learning lessons. Now, I would like to request Dr. Girish Babu K, sir, to say a few words on this occasion. Please, sir.
Good morning, everyone. Good morning, Dr. Chatterjee, sir.
Good morning.
It is immense pleasure for us to host you, and a scientist of your level, your caliber. It is really a great pleasure for us to host you on the webinar conducted by the Department of Life Sciences under the Applied Sciences. Sir, Parallel University, as such, is one of the fastest growing university among the private deemed university sectors. We have started our UG and PG courses since 2015, and within a span of 6 to 7 years, we have grown in a number of increasing the faculties and offering the different kinds of the courses for the students also. We have a beautiful, the output, I mean, like outside as such, for all the different colleges what we have somewhere around 34 different faculties operating under the Parallel University, and the Department of Life Sciences is also one of the oldest. Since the beginning of Parallel University we are there. We have that habit of inviting the most eminent scientists on our platform, so that our students and the other scientific community are going to be equally benefited. Listening to you and the people like you itself is going to be a great enlightenment for everyone, for us. So it is a great pleasure for us to have you here, and that too very interestingly, looking at the topic that the social language of the bacteria, speak or not speak, is really, really an interesting kind of a thing. We many a times being teachers, when we would be talking to the students, we always talk that we do have a communication, but at the same time there are plenty of organisms who don't speak like us, but they still have a fantastic and foolproof kind of the networking among themselves. And it is going to be a great opportunity for us and our students to listen to you. And I welcome you on the behalf of Parallel University and the management and the Faculty of Applied Sciences here. And we are very eager to listen to you, sir. Thank you so much for coming over here, and please enlighten us with your talk. Thank you.
Thank you so much, sir, for your kind words. Our eminent speaker is one of such personality who had many theories practically possible. Although an honorable speaker does not need any introduction, as he himself is an epitome of inspiration and proud of the country. Let me take this opportunity to introduce him. Professor Subhadip Chatterjee, sir, is an Indian molecular biologist and staff scientist at CDFD and member of Guha Research Conference. He did his doctoral studies from the laboratory of Ramesh Venkata Sonti, sir, of CCMB, Hyderabad. His postdoctoral work was at the laboratory of Stephen T. Lindbergh at the University of California, Berkeley. He is known to have carried out extensive research on plant microbe interaction system and has published a number of articles in high impact factor journals. He is an elected fellow of Indian National Science Academy, National Academy of Science, The Department of Biotechnology of Government of India awarded him fellowship and then the National Bioscience Award for Career Development and Innovation Young Biotechnologist Award. Anindi Kumar Ghosh Memorial Award was awarded to him in 1999. Professor Chatterjee was awarded the most prestigious Shanti Swarup Bhatnagar Prize for Science and Technology, the highest scientist award in India for the year 2020 in the biological science category by the hands of President of India. I feel privileged to invite Professor Subhadip Chatterjee sir to deliver his talk and enlighten us with his knowledge. Please, sir.
So, thank you on uh, on this occasion of this webinar. So, thanks, Dr. Dwani and Padhay for inviting me to deliver the talk. And I'm thankful to Parul University for inviting me to deliver the talk. So as you have now known that the today's my talk will be understanding the social language of bacteria, speak or not to speak. So I chose this topic because this is one of the very interesting field in which you will I will go through the social secret of bacteria, like how a tiny organism can have developed a very sophisticated language system, and how they utilize this language system to adjust to the changing environment. And sometimes they, like humans, they prefer to speak, and sometimes they don't prefer to speak based on their advantage. So I have divided my talk into three parts. In the first part, I will introduce you to the language system, like how language has been evolved in animal system, and how bacteria has evolved a sophisticated language system, which is called as quorum sensing. It is a chemical sensing and the chemical communication, which it has developed in the form of a language. And in the third part, I will talk about how it does this communication and what are the advantages of it to do the communication, and how it helps to thrive in the environment. And also it has a very big relevance in host pathogen interaction, because you have seen number of now bacterial diseases are multi-drug resistance, and how they develop this thing, the high pathogenicity causing harm to the human as well as animals and plants, and how they utilize this language system to cause disease and colonize in the host, and how they counter the language system so that it doesn't become disadvantage for them. So if you see in the evolution, the response by communication is one of the key living features of a living organism. So living organism has been characterized based on certain characters such as growth, they can reproduce, they pass on their hereditary material, they undergo stable homeostasis. That means they regulate metabolism and all the body reactions in a very regulated way. And they have a lot of cellular structures. But one key feature is the response. So all living organisms respond to the change in the environment. For example, if there is a fire, animals or other living organisms respond. They go away from the fire. They can sense it. And in this response, communication plays a very important part in alerting themselves and also their fellow mates. If you see, evolution of communication and language is very, as I told you, very critical for all living organisms. So the language has been developed from a very ancient to a very sophisticated way. And people, for example, the English language or the other languages, people compare it with the human evolution. As the are evolving, the language is also evolving. And they argue that the key factor in biological evolution, like natural selection, also happens in the language. For example, if you go within a region or a sub-region, the language changes, the dialects changes, and that is all because of the adaptation. So the language has been constantly evolving. Other than language, there are different mode of communication in different living organisms. As in the introduction, we have been told that different organisms communicate with different methods. For example, you might have seen fireflies, they glow in a particular pattern, and that scientists have identified that they emit a particular light at a particular frequency, and they give a signal to for the mating. Like for example, dance of the peacock or cobra movements. Like elephants do a lot of this sound, like very high frequency sounds. Whether the humans cannot listen, but it can be listened to a other herd, which is 2, 3, 10 kilometer away from the location. The most beautiful example of language is the dancing language of the honeybees. You might have seen honeybees. They have a dancing language. So by their dance ritual, they communicate to their fellow mates where the good flowers are. So where the good honey, good nectar they can get. So there are different forms of language. They use either audio visual, auditory, or based on the touch or chemicals. So the question came sometime back in the 15, 20 years back, that do unicellular solitary living microbes, which are such as bacteria, which are so tiny in nature that you have to see them in a electron microscope with a 1 lakh or 50,000 time amplification, magnification. So do these bacteria also have a social structure and communication method? So basically the question was whether the communication system is only developed in higher animals, and whether the communication system is a mode of any communication for the lower organisms such as bacteria. So soon people started discovering that bacteria not only always live in a solitary lifestyle, but often in the natural environment also. They form big colonies or structures which are called a biofilm. This is a confocal microscopic picture of a biofilm formed in a lab we have taken. And you can see the peaks. These peaks are nothing but buildings which have been made by millions of bacteria, and they form a housing society like colony. And this is a very communicative colony because they know what the neighbors are doing. And so these kind of structures you also find in natural environment. And a field of science which was developed, which was called as socio-microbiology, to study the scientific biology of the social life of bacteria or microbes, called socio-microbiology. And how the bacteria do the social sensing. So in prokaryotes, like such as bacteria, there is a phenomena called as quorum sensing, which is also called a density dependent signaling. So as the bacteria grows from a low number to a high number, they secrete chemical molecules in a density dependent fashion. So this is very important. So the secretion of the signaling molecules is a very density dependent process. As you see that the bacteria, when they are less in number, the amount of the chemical signal which is they secrete is also less. But when they grow at a particular level, the particular threshold has been reached, this concentration of chemical molecule also increases in the environment, and that gives a signal to the bacteria and tells the bacteria how many you are. So basically it is a counting procedure of the bacteria by counting the number of molecules of the signaling molecule which they secrete. They correlate that how many they are, whether they are in a group or a single or a small group. So this transition between a low cell density to a high cell density, so bacteria can count and recognize each other by their language. And this is an example of how they are talking with each other. So this is a form of language which you cannot listen, but you cannot see, but you can see them. So what you do, since I told you that these are chemical molecules and you can detect these chemical molecules, so we use lot of biosensors which can detect the chemical molecules. So for example, this is a plate in which full of bacteria is there and they are fluorescing green because they are able to detect the chemical molecule and they are glowing. So, and this is a plate full of bacteria. You see the bacteria, you can see, but they are not glowing. So they are not able to speak. So basically, this is a mutated bacteria in which they are unable to talk with each other, which we artificially made. But in the natural way, the bacteria loves to talk with each other. So what are their language molecules? So bacterial language molecules, as like human language system, it is very complex and there are many different bacterial language molecules. The most widely studied is this family of language molecule, which is called as N-acyl homoserine lactone. So this has a lactone ring which is common and different R groups are added. It is very similar to the analogy of the language such as Sanskrit. So Sanskrit is the base of many Indian languages. Similarly, lactone is the base of this homoserine lactone mediated communication, and different bacteria like pathogens and other environmental bacteria adds on different R groups, and these are different dialects, like for example Bengali, English, Hindi. So in our lab, which you will see in the next couple of slides, that we work on this family of signaling molecule, which we call a DSF, diffusible signal factor. It is a fatty acid like signaling molecule which are made by a large group of bacteria, which are very important economically because they cause lot of diseases in plants, and also many animal pathogens also make this family of signaling molecule. Like this long chain signaling molecules, many bacteria also make peptide like signaling molecules. And the interesting part is that, as I was telling you, these bacteria are very clever. So they not only, some bacteria can speak in one language, some are bilingual or trilingual also. For example, this is an example of Vibrio cholera, which causes cholera in humans. So these bacteria can communicate with two different signaling molecules. So this long chain signaling molecule, which I showed you earlier slide, that acyl homoserine lactone, is for communication among themselves. That is the private communication. The other language which it produces, which is borofuranol, it is for communicating with the other bacteria which are there in the gut. So communication with the neighbors. So very similar to a house situation where we speak with our native language, and if some guests come, we speak in a universal language. And there are advantages that many of the secrets which you speak in your mother tongue cannot be heard by the outsider. And this is the logic. And many bacteria are very clever. They don't waste their energy by speaking. So what they do is that they listen to the language of the other bacteria. For example, this bacteria, they cannot speak. That means they cannot produce the language molecule, but they have receptors, which are the ears of the bacteria, which they can listen to the language molecules which are produced by the other bacteria. So this is to adjust or modulate their own behavior based on your neighbors. Very similar to a old cartoon of Noisy Neighborhood, in which if a neighbors come in your locality, so the neighbors are more curious about the newcomers. Rather than interacting with them, they try to listen how they are. Like they have very similar analogy to this non-speaking bacteria, but they can listen to other bacteria. Now the main question arises like why this bacteria is doing so much of this, investing a lot of energy and mechanism for communication. So what is their advantage in having communication? So I will give you a very simplified example that you can which has an analogy to the quorum sensing. Suppose there is a single bacteria which is firing bullets to break the wall. The wall, you imagine the host barrier. So if there is one bacteria, the firing power of that bacteria is so less that if they fire bullets, it will be not able to break the wall. But when the bacteria is large in number and if they fire together, then there is a great chance that the wall will break. So this is the whole concept of quorum sensing. So that tells that there are certain tasks or job which gets the maximum benefit when you are doing it together. When you are alone, you can do the job, but the benefits will be less. That is the whole concept of quorum sensing. So therefore, bacteria synchronizes doing certain tasks when only at high cell density and not at low cell density to conserve energy of wastage of energy, because if they do the job alone, the benefits will be not that great. So in our lab, as I was telling you, we work on the quorum sensing signaling molecule DSF, and the bacteria which we work on is called as Xanthomonas, which is a very important group of plant pathogens. They causes more than 350 different plant diseases. So this is how the bacteria looks on a laboratory medium. It forms a yellow color colony, and the glossy, shiny thing which you see on the colony is a lot of polysaccharide it makes. And as I told you, it has a very diverse host range. 350 different plant diseases are there, very economically important, such as they cause disease in rice, many vegetables such as cabbage, fruits. So we particularly concentrate on the rice pathogens and the cabbage pathogen. So rice, because why? Because it's a staple food of India, and lot of the area in India is dependent on rice cultivation. And this bacteria causes a very bad disease in rice, which is called a bacterial leaf blight. And you can see this is a leaf which is infected with this bacteria, and the whole rice plant dies within 10 to 15 days of infection. So we started working on how this bacteria causes disease on rice. So what we discovered that this bacteria makes a signaling molecule, as I told you, that quorum sensing signaling molecule, which is called a DSF, and DSF is required for virulence in Xanthomonas. That means this making of chemical is very important to cause disease. For example, this is a rice leaf which is infected with a wild type bacteria, which is able to make the chemical. But if you infect a bacteria which is not able to make the chemical, it is not able to cause disease. But when you again put back the chemical inside the bacteria by either artificially or by genetic manipulation, it is again able to cause disease. So this is was a very interesting biological question: how a language molecule will regulate its pathogenicity and virulence. So this is how the bacteria looks inside the plant. So this is a very high resolution confocal micrograph in which you see the red color pipe-like structure, which are the xylem vessel of the plant. So this bacteria colonizes inside the xylem vessel, and the green color is the labeled bacteria. So these are all live bacteria. And we started observing that this bacteria forms a very nice biofilm or multicellular community-like structure inside the xylem vessel. And when we studied it more deep, I am not going much into detail. A lot of work has been went on. So this is how the model system, if you see, if you imagine the life of the bacteria, suppose it lands up on the leaf surface, the green part is a cartoon which is depicting the leaf surface. So it has to enter through the doors or the openings. But if you compare the size of the bacteria, it is too less, too small, and it will require a lifetime of its lifetime to reach at the spot. So therefore, it should have activated a chemotaxis system or a guided modality towards the door. And this is how the quorum sensing plays a role. At low cell density, what we discovered is that when the bacteria is less in number, it gives a signal to the bacteria, now you activate your guiding system so that you can enter. And once they enter, they start secreting enzymes and effectors which can now damage the plant cell and release nutrients. And with the released nutrients, they start growing in more in numbers. And when it reaches a threshold, they forms a protective macrocolony or a community. And this community helps them to protect them against the defense responses and the harmful harsh environment of the plant. And also they activate lot of nutrient uptake systems such as iron. And this transition from a solitary lifestyle to the biofilm lifestyle, the quorum sensing mediated by DSF plays a very important role, because at high cell density, they makes adhesins which are attachment factors when only in high cell density. So they don't make attachment factor here at this stage, because this will be very disadvantage for the bacteria, because they will stick at the very early stage and they will be not able to move and colonize. This is a molecular model. I will not tell you to go through it. It's a very complex gene regulatory circuit which we have come across after a lot of work in the lab. But what the take home message I wanted to tell you is that bacteria utilizes a very complex and sophisticated information processing system by quorum sensing to perform various tasks. So there are a number of regulators and transcription factors which play around in sensing the signal, also other environmental signal, and they integrate the information to regulate expression of thousands of genes in a density dependent fashion to regulate the bacterial social behavior like planktonic to biofilm transition. This is overall model which I was telling you, that at low cell density, the signal of the quorum sensing activates the chemotaxis system, then it goes and colonizes and start damaging the host cell to get nutrients to become more in number. And when they become much more in number, they form a biofilm. And we also discovered another factor, that this stage is also very deleterious for a long term, because again the bacteria has to move from this place to a new place. And what triggers is that a switch we identified is called a reversible non-genetic heterogeneity. That I will tell you very briefly now. So now I will tell you about the disadvantage of talking. So the talking, as I was telling you, is a double-edged sword. There are very many advantages to talk, to communicate, and to synchronize, but there are also disadvantages. So this main disadvantage is that cheating. So for example, in a group of bacteria, if you see this in gray color bacteria, they are talking with each other. But in that population, if somebody starts stop to talk, and they take all the advantage of the talking bacteria. Very similar to an examination in which these fellows have worked hard and for the examination, and they are writing, and there are certain cheaters which has not worked hard, but they take advantage of the hardworking people. So in this way, the whole system will collapse, because in next time the performers may not also perform, and they start cheating, and whole system will collapse. So this is what we call a altruism. Altruism is the benefit for the whole society, and that brings about conflict also. The second disadvantage of talking is that you can alert by too much talking, you can alert the host defense system. For example, in this cartoon, if the thieves are talking too in a loud voice with each other, they can alert the security system. So how the bacteria counters these two problems? So we identified that the bacteria has a division of labor in which it maintains both talking and non-talking population. Talking population for doing the task and non-talking population for doing the task which has to be done at low cell density. So this gives them the advantage of both working, so they give the advantage of both talking as well as and non-talking. And the system will not collapse because there is a tight regulation of the talking and non-talking population. So this is what we have made the model, that the biofilm formation and how they undergo transition. So now I will very quickly, within 2 minutes, I will finish how we can apply what we have learned from basic research, that whether we can interfere with the bacterial quorum sensing signaling molecule to make our life better. So what we call of a social drugs as an antimicrobial agent, which is an alternative to the chemical therapy or the antibiotics. So the idea is either you stop the talking of the bacteria so that they can communicate, they cannot communicate. The other strategy is to produce the chemical or the communication in so high in amount that it will act as a noise pollution. Very similar to if you are in a very crowded and noisy place, you will be not able to communicate with your fellow mate. So we did both transgenic and non-transgenic approach. So what we did in the transgenic approach is that we transferred the whole bacterial quorum sensing system in the plant. And now these plants started producing the bacterial signaling molecules. So the idea was that if this plant which is producing the bacterial signaling molecule, when a natural bacteria will infect this plant, it will sense that we are too high in number, we should not infect, we should form a biofilm at the point of entry itself, and the disease will stop. As you can see, this is the results which we got from the transgenic plants. So this is the y-axis, you can see the disease index. So you can see the wild type is heavily susceptible for disease, and you can see all the transgenic lines are very susceptible to, very resistant to the disease. We did another approach also to artificially produce the quorum sensing signaling molecule by biotechnological method in other bacteria, and we, instead of using transgenic, you can use it as a spray to spike the uninfected plant, so that when the infection actually happens, the bacteria is fooled to sense that you are too high in number, and then you stop the infection. So this is our laboratory complex in Hyderabad, CDFD, and all the people in the yellow color are the current lab members who are working on different projects of social sharing and social microbiology. These all people in the green are postdocs and graduate students who are now have done a tremendous amount of work in our lab, and they are now postdocs or faculty in the other places. And I would like to thank all the granting agencies for providing support for the research. Thank you.
Thank you so much, sir. Thank you so much. I would like to request you to answer the questions if you allow. There are few students and they are having questions related to the same.
Yeah, sure.
Shall I continue, sir?
Yeah.
Yes, sir. So first question is that Do you like to know about working on discovering reversible non-genetic heterogeny in
Heterogeneity. Yeah.
bacteria?
So I think your voice is not clear. It is breaking.
Hello, sir. Yes, sir. Am I audible, sir?
Yeah, now it's audible. Yeah.
Yes, sir. So I repeat the question again, sir. Sir, student has asked that we would like to know about your research experience by working on discovering reversible non-genetic heterogeneity
Okay,
in
okay, I
bacterial and
understood.
social.
Understand the question. So reversible non-genetic heterogeneity happened by a serendipitous experiment in which what we observed is that we, as I told you in the beginning of my talk, we use a lot of biosensors to detect the quorum synthesis signaling molecule. So when we started looking in a single cell level in this bacteria, we saw a lot of cells, significant proportion of cell, around 10 to 15%, they are not responding to the quorum. So then we found out that this non-responding quorum is a stochastic reversible population, which again in the next generation give rise to the talking and non-talking population in 1:4 ratio. And in that way, those who are perform the social task. Those who are non-sensing or non-performing, they do the task which they have to do when they are less in.
There is one more question.
Yeah.
Yes, sir. Sir, what is your current research in plant microbe interaction at CDFD?
So this is the one which I told you in the talk. So our main research, how the quorum sensing regulates virulence.
Respected sir, I think there is some noise
Yeah, a
coming
lot of
from your
background
side.
noise is there.
Yes, sir. Sir, can you answer the same?
Yeah, the answer is that we work on three aspects in CDFD. One is the quorum sensing of Xanthomonas, how it is important for its pathogenicity, and how the plants can detect the quorum sensing signal molecule and mount a defense response. And the second aspect, the second aspect is we work on the molecular mechanism of how this sensing is controlled and how the signal transaction process is going on. And the third aspect, we work on the plant aspect, like how the plant is responding to the bacterial quorum sensing signaling molecule. And the fourth part, we work on like how this bacteria actually hijacks the nutrient system in the plant.
So, sir, there is one more question. Sir, can you tell us that what are the applications of the quorum sensing other than that what we had discussed?
So I told you about the applications of quorum sensing in the last slides. So there is a big research which is going on social drugs. So like antibiotics, it is very easy to get resistance because you have seen nowadays like bacteria are not going by heavy dose of antibiotics also. That is because of mutation. Because the mutation in the drug, this targets was causing them insensitive. But however, if you target the quorum sensing, because it's a whole system you are targeting, so getting a resistance is very less frequency is there to get mutation in many places instead of one. So therefore, people are using quorum sensing interference as a strategy to prevent very bad diseases in both human and plants.
Thank you, sir. There is my advice to give the researchers if they want to work on the quorum sensing.
Pardon?
Sir, there are some research scholars who are listening you. They are asking that what piece of advice would you like to give them if they want to work on the quorum sensing project?
So the piece of advice is that you study the basic microbiology properly and read very interesting papers, how this quorum sensing was discovered and what is its role, as I was telling you. There are many interesting literatures, etc. is there. There are few people working in India, abroad. So you try to read and try to understand and interact with these people, and you will much better, you will know this subject.
Thank you so much, sir.
Thank you.
Uh,
Yeah.
I would like to thank and I extend the gratitude to the Dr. Subhadip sir for accepting our invitation, and extremely thankful that take out few minutes for us from his busy schedule and for this knowledgeable session. So thank you so much, sir, on behalf of entire Parul University, and thank you so much all the faculty members and students, those who are listening us. Thank you so much,
Thank you.
sir.
Thank you very much. Wish you all the best. Thank you.
Thank you.
So can I leave now the meeting?
Yes, sir.
Thank you.
Thank you so much.